So, welcome to this April edition of the HUPO ECR and YPIC webinar series. Today we have the honor of having the winners from last year's 3-minute thesis competition that will present their work in a more extended format compared to last year's competition. So they will have 10 minutes with some additional take-home messages from last year's competition each. And then afterwards, we will have some Q&A session where you're allowed to ask questions about the competition or maybe about your work. So before we get started, I will just share a little something about this year's competition. So as last year, we will be hosting the 3-minute thesis competition during this year's Congress in Dresden. It's presented in collaboration with the Human Proteome Project. And in order to participate, you are this year supposed to submit the lay abstract, which will sort of explain your PhD thesis in a shorter format that is more catering towards a general population rather than a scientifically well informed population. So the idea is to sort of then present your thesis with only one slide without any graphics or moving animations. And the winner of this competition, which will be decided by a set of panelists from well-known scientists within the field, will receive a cash prize of $500, and the 2 runner-ups will receive a cash prize of $300. And if you're interested in participating in the competition, the submission deadline is the same as for all of the other abstracts at this year's Congress, so the 31st of May. And in order to be eligible, you will have to have not defended your thesis upon applying for the competition. And I also just want to highlight the 2 other competitions that might be of interest to all you early career research, and that's the manuscript competition, which is our biggest competition, and where we sort of end with the proteomics highlight of the year. So you get the full presentation slot at this year's Congress. And also we have the poster competition, which has been our most long-standing ECR competition together with the manuscript competition. So with that, I will start with presenting our first speaker. And our first speaker is Dina Schuster, who, if I'm not mistaken, has just defended her thesis a few months ago. She did her bachelor's
in June.
Awesome. She did her bachelor's and master's degree in chemistry at the University of Vienna, where she introduced and started working in the field of mass spectrometry. In 2019, she moved to Switzerland to pursue a PhD at ETH Zurich within the group of Volodymyr Korokov and Paolo Picotti. During that time, she worked on projects that interface the structural biology and structural proteomics with focus on membrane proteins. After finishing her PhD in 2023, she moved to the Netherlands to a postdoc position in the group of Albert Heck at Utrecht University. Right now, she's working with instrument development projects together with Brucker, where she's establishing new intact protein naive and top-down MS workflows for the analysis of antibody repertoires. So with that, I'll hand over to you, Dina.
Thank you for this nice introduction, and thank you also for having me here today. I'm happy to tell you a bit about my PhD work. I haven't talked about it in a few months, so it's good to get back into this. My PhD was focused on membrane proteins. I did establish a workflow, an integrative structural proteomics-based workflow to study the regulation of different membrane proteins. Today, I will focus on adenylyl cyclases just to illustrate what I did. Membrane proteins are located at all kinds of different membranes. I was focused on membrane proteins located at the plasma membrane. The plasma membrane you might be familiar with is a lipid bilayer that acts as a physical barrier to protect the cell from outside influences. Inside the plasma membrane, we can have different proteins that can carry out various functions. We can have proteins that sense different small molecules or different signals from the outside. We can have proteins that aid in transport of small molecules, for example. We can also have proteins in there that help in signal transduction, or we can have proteins in there that help with cell-cell communication. Membrane proteins themselves, they make up a large part of the human proteome, roughly 20 to 30%, and they make up 50% of the plasma membrane mass. So despite their large representation in the proteome, they're mostly uncharacterized. They're still the largest group of therapeutic targets, but there is still a lot to understand for those proteins. And they remain difficult to study because of their high hydrophobicity, their low abundance, and difficulties to purify those proteins. Just like other proteins, membrane proteins are biopolymers that are composed of amino acids. And those amino acids, they branch together and they form different secondary structures such as alpha helices, beta strands, and other structures. And these structures, these alpha helices, for example, they fold into larger 3D structures that you might be familiar with seeing them here, either as the structural representation or as a surface representation on the right. And these structures, they don't show you the full picture. In these structures, there's also unstructured regions, flexible or highly disordered regions. And we were actually very interested in those disordered regions because up until recently, the main paradigm in structural biology or biochemistry was the structure function paradigm that says that a protein sequence determines the structure and that determines the function of the protein, which largely holds true. But in recent years, there have been more and more studies that kind of coined this disorder function paradigm that states that not only structure has a function, but also disorder can have a function. And those functions can be varied. They can be the regulation of the protein. These disordered regions can be involved in interactions. They can act as chaperones. They can harbor post-translational modifications, or they can act as linkers, for example. And in membrane proteins, we have many of those disordered regions. Actually, roughly 70% of all membrane proteins involved in signaling have such long disordered regions. And these regions are difficult to study with traditional structural biology tools, such as cryo-EM, for example. If you look at the cryo-EM structure of a protein, or of this protein here in particular, you will see that a big part of it is missing. So if we predict the structure with AlphaFold, there are many more loops that you would not be able to see with cryo-EM. And those loops are the flexible disordered regions that move around and that cannot be resolved with cryo-electron microscopy. So the aims for my PhD were basically to develop a workflow that helps us study those membrane proteins and their flexible domains without the need for purification, because that is difficult and a lot of work. And to then apply this workflow to understand the regulation of different kinds of membrane proteins. The membrane protein I will talk about today, this was one of the proteins I focused on in my PhD, is adenylyl cyclase 8. And for this project, I was working closely together with a postdoc in one of my labs, Basavaj here, shown at the top. AC8 is a signal translator and signal transductor located at the plasma membrane of cells. You can see it here in the middle in red. This protein is mainly found in the brain, where it is responsible for learning and memory. And this protein senses signals. So the GPCR senses signals from the outside that are then translated by this adenylyl cyclases. In turn, it catalyzes the conversion of ATP to cyclic AMP, a signaling molecule that triggers all kinds of events, such as, for example, phosphorylation cascades. And this protein, we did not really know how it was regulated, so we wanted to know how exactly is it regulated, where do these interactors bind, and what is the role of its flexible regions. And to understand this, we first tried to solve the structure with cryo-electron microscopy, but again, to not really our surprise, we did see the structured domains of the protein, but we did not see the flexible regions. The flexible regions here are the N-terminus, the C1b domain, and the C-terminus of the protein. We wanted to know one specific interaction. We wanted to know more about the inhibitory interaction with the G protein, G beta gamma, mainly because we could not study this interaction with cryo-electron microscopy at all. So if we put both of those onto EM grids, we would just see AC8, but there would be no evidence of the existence of G beta gamma. So we were wondering, how can we actually study those interactions with the tools that we have available? And one method that had recently been published, but only used so far for soluble proteins, is limited proteolysis coupled mass spectrometry. So I adapted the workflow to be used for membrane proteins. And mainly what you do in limited proteolysis coupled mass spectrometry, or LipMS, is that you digest your protein of interest with an unspecific protease. You generate structure-specific fragments that you then denature and digest with trypsin. Now, if you were to study a protein-protein interaction, you would do the same thing, add your interactor of interest, generate those fragments, analyze it with mass spectrometry, and compare this to the protein by itself. With some computational help, you can then generate these sequence-specific barcodes or protease accessibility barcodes that will tell you which regions of the protein are less accessible to the protease and therefore likely to be the interaction site. This is a very great method as it can help you access or assess accessibility changes directly in the lipid environment. We use membrane suspensions to probe these interactions, and you don't need to purify the membrane protein. Unfortunately, with the method, as no method is perfect, you will not be able to detect transmembrane domains. You will not get any information on proximity, so how close are your interactors? You won't be able to assess this. And you also cannot distinguish interaction sites from conformational changes. But because we wanted to do this, we decided to combine this workflow with cross-linking mass spectrometry. So we looked at the same interactions with cross-linking mass spectrometry, where we covalently link specific residues in our protein complex. We digest it again with a protease, enrich for cross-linked peptides, analyze this with mass spectrometry, and then evaluate this to figure out more about the interaction site. And in this way, we could obtain proximity information on our two interacting proteins, but this is a bit difficult to perform in complex systems. So here we had to use purified protein. The results that we got out in the end, combining cryo-EM, LipMS, and cross-linking MS were as follows. With cryo-EM, we could see the structured domains of the protein, but not the flexible regions. With LipMS, we were able to determine that actually this G protein, G beta gamma, interacts with the N terminus, the C1b, and the C terminus, so all three flexible regions. With cross-linking mass spectrometry, then, we could pinpoint interaction sites, and we could use these interaction sites for docking studies where we were actually able to model the protein complex of those two interactors together. And what was very interesting to us is that when we had modeled this structure, at roughly the same time, another group published a structure of a different adenylyl cyclase isoform with the same interactor. And we could see that the structure we obtained from mass spectrometry results was actually very similar to the structure obtained with cryo-EM. Here you can also see that not the full part of the protein is resolved, as this is likely also very flexible. So what we could conclude from these results was that these flexible regions are indeed very crucial for the interaction with G beta gamma and likely also involved in the inhibition of our protein. We could see that G beta gamma interacts with one particular flexible region, C1b, and we could use these residues for docking studies. So to conclude, with cryo-EM, we can get high-resolution structures. With LipMS, we can get protease accessibility information. Cross-linking mass spectrometry, we can get proximity information. And through the combination of all those three techniques, we could obtain more information on flexible domains, see that they are very important for the regulation of different membrane proteins. We could use LipMS to study membrane protein interactions without protein purification. And LipMS and cross-linking give us complementary information that we did not see with cryo-electron microscopy. So with this, we were able to generate different hypotheses and structural models for various different membrane proteins. This is also what I presented at the 3-minute thesis competition. I did not know that Andreas would give a bit of a recap of the thesis competition. So here is a bit of repetition of what this actually is. So I participated last year in Korea, where to participate in the 3-minute thesis competition, I submitted a lay abstract describing my work in very easy terms. This was a great exercise for me as it really helped me to boil down what is actually important in my research and to be able, or it helped me to be able to explain my work in simple words to people who might not be within the same research field. And you are eligible to do this if you have not defended your thesis yet at the time of abstract submission. What do you need to prepare? You need to prepare one static slide and then a 3-minute presentation of your work. For me, what is in it for you? For me, this personally was a very good experience. I got to meet very nice other PhD students who were at the same career stage that I was in at that time. I learned a lot about different kinds of research that I had not been familiar with. I made some friends at the conference. I learned more about their PhD theses. So also socially, this was a nice experience. You really practice your presentation skills in that competition. As you only have a very limited amount of time, you really need to boil down the information to the most crucial part. And it helped me, and it will help you to prioritize content. So what did I present during this competition? I will show you the slide that I was showing, and I hope that right now you're a bit more familiar with my slide and you know what this slide is about. So I was again talking about membrane proteins. Why would we like to study them? What would we really like to study? Flexible regions. Why are we interested in those? How do we go about this? And then what were the most important results of my work? So with this, I would like to thank you for your attention, but I would also like to thank everyone who was involved in my PhD. There were quite many people who helped me with the work, and I could not have gone to HUPO without them, and I could not have presented this work without their help. Thank you very much.
Thank you so much, Lina, for a stellar presentation. And I guess in order to keep time and then save the questions for later, we will go ahead with the next speaker, which is Janaina Silva. And Janaina has a degree in biotechnology engineering and a master's degree in biosciences/biotechnology, and is currently studying the last year of her doctorate under the guidance of Professor Dr. Giuseppe Palmisiano. She has experience in study of post-translational modifications through mass spectrometry, mainly focused on study of arginylation and N-glycosylation of proteins in cancer. She has extensive experience in analyzing integrating omics data, including genomics, transcriptomics, and proteomics and metabolomics. She also applies molecular and cellular biology techniques and tools in conjunction with mass spectrometry to elucidate signaling pathways and crosstalk between cross-translational modifications. And with that, I will hand over to you, Janaina.
Good. Is everything works?
Yes, we can see your presentation.
Good. So hi, everyone. Thank you for the invitation and thank you for the introduction. And I'm going to show some data that I shared in the last HUPO 3-minute Thesis Competition. And the topic of my presentation is protein arginylation. So before I start with the results, I think it is important to show you how protein arginylation works, because in the last conference, I noticed that protein arginylation is not so famous or well explored. And so protein arginylation is a post-translational modification involved in the N-digron pathway.
Sorry, Janaina, we don't see these slides right now.
Can you see?
We see the PowerPoint window, but not the presentation slides.
Sorry, I don't know why.
If you try resharing, maybe.
I'm sharing. Now?
Now we see it. Perfect.
Good. So now I can. So protein arginylation is involved in the N-digron pathway, and this pathway basically say to us that the amino acid exposed at the N terminus of a protein is responsible for the half-life of this protein. So when we have cysteine or glutamic and aspartic acid exposed at the N-terminus of the protein, this protein can be arginylated by the H1 enzyme. So basically this enzyme transfers an arginine to the N-terminus of the protein, and this protein is then ubiquitinated and degraded by the proteasome or autophagolysosome. So we did some bioinformatics analysis and we found that almost 4% of the human proteome can be arginylated. So almost 4% of the human proteome has a cysteine or glutamic or aspartic acid exposed at the N-terminus. But it's curious that nowadays we have only 60 arginylated proteins well characterized. So we have a huge gap in this topic. So I started my PhD during the SARS-CoV-2, the pandemic, and we did a study, a proteomic study, when we analyzed the infected cells, and we found that when we infected cells with the virus, we have an increase in ER stress. And we know that protein arginylation is closely related with stress response. So we ask ourselves, what is the relationship of the protein arginylation and the ER stress induced by the SARS-CoV-2 infection? So we start exploring public-omic data at transcript and protein level, and we found a general dysregulation of the endogram pathway. We focus mainly on the dysregulation of H1. So basically, we found that the H1 protein is upregulated in infected cells. And we also performed our own experiments. So we infected 2 different kinds of cells, and we did a time course infection, and we found a upregulation of H1. When we spike transfected the cells, we also found a upregulation of H1. And when we transfected the cells with the RBD region of the spike protein, we found that the Delta variant increased more the levels of H1. So using the public proteomics data, we selected a panel of 28 proteins, which is correlated with H1 levels, and we found that these proteins correlated with H1 is closely related to ER events like protein fold or unfolded protein bind or protein localization to ER. So basically, we conclude that this increased H1 expression during the SARS-CoV-2 infection is related with chaperones, protein fold, and endoplasmic reticulum. And it makes sense because we found that the protein is a chaperone, it's arginylated. So, for example, in calotroph cells, when we infected the cells, we have an increase in the levels of the arginylated portion of the BIP. And also, when we transfected the cell with spike glycoprotein, we have an increase in the arginylation of BIP. And it's really interesting because BIP is the main regulation of the UPR pathway. So basically, when the BP protein is binding in the UPR receptors, this pathway is not activated. So the structural change in the BIP and arginylated BIP is really important to activation and deactivation of UPR pathway. And here we can see clearly different changes in the structure when we have BIP and when we have BP protein. We can see change mainly in the KDL motif, and also in the molecular dynamics, we can see difference in the flexible of the arginylated and non-arginylated protein. So when we infect a cell with SARS-CoV-2, the levels of arginylation increase. So what happened when we decrease the levels? So we silence the H1, we silence arginylation by siRNA, and also we perform the chemical inhibition using tannic acid and mebromine. And we found that the decrease of arginylation also decrease the viral release, here by PCR, and here we can see by Western blot analysis. So we ask what is the mechanism involved in the reduction of the viral release? And to answer this question, we performed a global proteomic analysis in the silent cells. And we found a downregulation of N-linked glycosylation proteins and also in proteins involved in ER stress. So first, we validated the downregulation of the N-linked glycosylation machinery, and we found that the OST complex is downregulated. This complex is responsible to transfer a glycan to an nascent glycoprotein. So when we silence the cells, the glycan is not being transferred to the glycoprotein. And we also performed the glycoproteomic analysis, and we found a downregulation of the glycopeptides in the silence group. And these, the glycopeptides come from receptor proteins. This is really interesting. So to test our hypothesis, we first infected cells and found an upregulation of OST proteins. This is not new because other groups have published this. So when we infected a cell with SARS-CoV-2, we induce the N-glycosylation machinery. But when we infected silent cells, we can see this increase. So the silent cell has some issues to transfer the glycan to the nascent protein. So we did a lectin blot using concavalin. So we can see here also a downregulation of concavalin A in the silence group. And also we can see a downregulation of the RBD region of the spike protein from the virus. So our global proteomic analysis also showed downregulation of ER stress and UPR pathway. And we also validated this data. So when I silence H1, we also silence the UPR activation. And the opposite also is true. When I induce the stress in a cell, I increase the levels of H1. So there's a relationship between H1 and UPR activation. So based on this, we silence the cell so the cell can respond to a stress, and we induce a stress in the cells to check. When we induce stress by serial deprivation and TAP-C-guarding treatment, we found that the silent cell is dying. So we was thinking about this, but now not in virus infection, but in a cancer context, because it's interesting. If I silence H1, the cell can handle it with stress conditions. It makes sense if I silence the cells. In a cell, in cancer cells, I can kill these cells. So we performed a study using GBM cell lines. So basically we overexpress and silence H1, the enzyme levels, but also the activity of the enzyme. And here we found that when we silence H1 and we induce a stress, for example, with the treatment of terlozolamide, the silent cell is dying. And probably because this cell can respond to stress. Also, we did a proliferation curve. So when I overexpress H1, the cells, the proliferation increase. But when I silence H1, the proliferation, we can see difference in the proliferation. We can see difference only when we induce stress. So to be sure that the proliferation and the response to stress pathway was regulated, we performed a transcriptomic analysis of cells overexpressing H1, and we observed upregulation of ER stress, of UPR pathway, and also in autophagy pathway. And we validated this in silence and overexpressing cells. So basically, When I increase the levels of H1, I change the levels of their generated BIP, and I can modulate the UPR pathway. So overexpressing H1, we have more activations of the ER stress and UPR. When I silence H1, I have a decrease in the UPR activation and ER stress. And as a downstream effects of the UPR, we have autophagy and apoptosis. And here we can see clearly that H1 levels induces the autophagy, and when we silence the H1 levels, we don't have an activation of autophagy. So maybe that's why the cell fitness increase and also the proliferation of the cancer cells increase. And one of the most interesting data are these results is reproducible in human samples. So we tested in GBM and normal controls, and we found activation of H1 in GBM samples. And by proteomic analysis, we can see here a clear separation between the GBM samples and control samples. And here this PCA was building using only the ND-GROM pathway proteins. And here we can see that the levels, high levels of H1 is related to low survival. So how to put all this data in a single slide and present in 3 minutes? It was a big challenge. I was nervous, to be honest, but we developed an approach, and this is my slide in the 3-minute thesis competition of the last year. And I will show you what we think to do this. So first, we think in a visual approach. So if the increased levels of the protein that I'm working is good for the virus is happy. So I put a happy face here. Also the size, if the increase of the levels of H1 is good for the tumor proliferation, I put a big tumor here comparing to a small tumor here. So during the presentation, we focus in present our results. We don't focus on how we discovered it or what kind of experiment we did to conclude this. We focus mainly on how we focus on what we discovered. So also we changed the colors to project to colors. We also use interesting words, for example, human samples or drug, terlozolamide. We use here a drug to represent. And to finish, we focus on what happened and why it happened, and don't explain in a deep way. So to finish, thank you for all my team, and thank you for the ECR for the invitation, and I hope this presentation helps someone in the next 3-minute thesis competition.
Thank you so much, Jenna. It was a really interesting breakdown of your presentation as well. So thank you for that. And I guess then we will continue with the last speaker of this session, which is Junyoung Yang, who is the graduate student at a researcher at Taipei Medical University in Taiwan. And she's currently working under the supervision of Dr. Yilin Tsai. And her main area of focus is glycoproteomics and biomarker investigation. Her current research project centers around the investigation of antibody glycosylation in the context of infectious diseases, such as COVID-19 and tuberculosis. So with that, I will hand over to you, Junyoung.
Hello. I'm sorry that my internet is not working very well, so I'd better not to turn on my camera. I apologize.
Okay.
So, can you see my screen now?
Yes, we can see your screen.
Okay, so thank you for the kind introduction and thank you for inviting me. My name is Yang Yunrong from Taiwan, and the topic I want to share with you today is distinctive antibody glycosylation profiles in ESRD patients with divergent immune responses to COVID-19 vaccination. So patients with end-stage renal disease, ESRD, are mostly immunocompromised. So they're particularly susceptible to COVID-19 infection, along with higher disease severity and higher mortality. So that's why vaccination is particularly critical to protect these people. However, they also experience weaker antibody responses toward COVID-19 vaccination. Clinical predictors of vaccine responses for ESRD patients include age, previous infection, immunosuppressive therapy, body mass index, and serum albumin level. However, how about the role of the antibody itself? So the protective function of antibodies is determined by various factors. One is the antibody levels, which is relatively easy to measure. And the FAB domain of antibodies is responsible for antigen recognition and neutralization. And the FC domain binds to the FC receptor on immune effector cells, then performs the FC effector functions. So the binding affinity and the effector functions are regulated by the selection of antibody isotypes and subclasses, and also by the glycosylation modifications. So previous studies have demonstrated that the antibody glycosylation is associated with COVID-19 infection, severity, vaccine response, and breakthrough infection after vaccination. However, these studies are mostly limited to IgG glycosylation only, and no studies are specific to the vulnerable ESRD patients. So here are our study aims. First is to establish a correlation between antibody glycosylation features and COVID-19 vaccine response diversity in ESRD patients. And second is to identify potential biomarkers that can predict vaccine responsiveness. So here's our workflow. We recruited ESRD patients in Taiwan and administered the AZ vaccine. 10 weeks later, we measured their COVID-19 specific antibody levels and divided them into responders with high antibody levels and non-responders with low antibody levels. The affinity beads were used to purify all classes of bulk immunoglobulins from the serum samples. And 2 enzymes were used to obtain the peptide and glycopeptide fragments for subsequent mass spectrometry analysis. And we used a triple quadrupole system to analyze our targets. So our study included not only all IgG subclasses from IgG1 to IgG4, but also IgA1 and 2 and IgM analysis. Each of them has their distinct unglycosylation sites. And the glycosylation features we analyzed include galactosylation, sialylation, fucosylation, bisection, high mannose, and hybrid type glycans. So we comprehensively analyzed the glycosylation profile, including 3 antibody isotypes, 6 antibody subclasses, 10 unglycosylation sites, 49 glycosylation traits from different subclasses or glycosylation sites in combination with 9 glycosylation features, and a total of 105 glycopeptides from IgG, IgA, and IgM. So here are the results. Out of the 49 glycosylation traits we analyzed, we identified 6 that were significantly different between responders and non-responders. In this specific glycosylation site of IgA, compared with non-responders, the responders showed lower digalactosylation, higher trigalactosylation, and higher sialylation levels. And in IgM, responders showed higher sialylation and fucosylation levels and lower levels of high mannose glycans. So a previous IgG glycosylation study reported that in the general population, not ESRD, people with a high antibody response after vaccination showed increased IgG galactosylation and sialylation. The authors suggested that these glycosylation changes might be protective against COVID-19. However, in our ESRD population, we didn't find a significant difference in these IgG glycosylation features between responders and non-responders. Instead, we found that the responders displayed this similar trend in IgA and IgM glycosylation features. This may be because ESRD patients often experience a delayed IgG response, so these changes are seen in IgA and IgM antibodies rather than IgG. Also, in the general population, people who were vaccinated showed increased IgG fucosylation. So similarly, in our ESRD population, responders displayed this similar trend in IgM antibodies rather than IgG. This may be also because of the delayed IgG response that is often observed in ESRD patients. And there is one previous study that focused on IgM glycosylation. They reported that increased IgM sialylation and decreased high mannose glycans are associated with severe COVID-19 disease. They also observed elevated IgM-dependent complement deposition in these severe COVID-19 patients. So in our ESRD population, the responders displayed this similar IgM glycosylation trends. Moreover, we found that in non-responders, the IgM sialylation levels positively correlated with anti-COVID-19 IgG levels. So a previous study also reported that elevation in IgG1 sialylation correlated with higher anti-COVID-19 IgG titers. So again, we found a similar result, but in IgM sialylation rather than IgG. And among the 105 antibody glycopeptides we analyzed, 10 glycopeptides were significantly different between responders and non-responders, including 3 IgG, 3 IgA, and 4 IgM glycopeptides. So these 10 glycopeptides were used for the ROC curve analysis. We can see that their discriminative performance between responders and non-responders achieved an area under the curve of 0.82, suggesting their good potential as biomarkers in predicting COVID-19 vaccine responsiveness. So here's a brief summary of our work. In ESRD patients, COVID-19 vaccine responders exhibited an antibody glycosylation profile similar to that of the general population who have a high vaccine response or severe COVID-19 disease. However, this similarity is observed in IgA and IgM antibodies rather than IgG. This may be explained by the delayed IgG response that is often observed in ESRD patients. Also, we identified 10 antibody glycopeptides that can effectively differentiate between responders and non-responders. So finally, in our future research, we will perform the COVID-19 specific antibody analysis regarding this topic. So as for my experiences preparing for the competition, same as previous speaker said, I think the most challenging part is the 3-minute time limit and that the target audience is the lay audience. So this means I have to explain a whole story in very simple language within a very short time limit and within one slide only. So personally, in my slide, I used a lot of cartoon illustrations to explain the research background. And also I used some very straightforward sentences to directly list our findings. So here are some of my experiences preparing for the competition. So I would like to thank my supervisor, Tai Ling, our collaborators, the NSTC funding, and also the HUPO ECR committee for this meaningful prize. Thank you.
For a fantastic presentation. We have some questions in the Q&A tab that are mainly focused on. I've seen that Diana has answered one already, but Janina, there is a question about what kind of methodology you used for the global proteomics. Was it DIA or DDA?
We use DA approach, and we perform a regular in-gel digestion, and for the glycoproteomics, we use PNGA's enzyme and Helic.
Thank you. And then we have one for Yun. Have you tried any antigen-specific enrichment of the antibodies, or are you just looking at the groups, I guess, of antibodies? And if you have done any antigen-specific enrichment, have you seen any unique patterns to those groups of antibodies?
Okay, thank you for your question. So far, we haven't tried any disease-specific antibody research, not yet. But we only look for bulk immunoglobulins, their glycosylation patterns. So it's our future work.
Perfect, thank you. So we had some questions as well. So we will, I guess, we'll go around in order here. I guess most of you have already answered what's the most difficult part about summarizing such a vast project in 3 minutes. But I have like 2 questions, I guess, specifically for Dina and for Janina, who are like so close to finishing your PhD as well. Have like this way of thinking about your thesis helped in sort of communicating, or at least at my university, we used to have this like sort of more popular science approach to explaining your thesis as like the abstract. So has this way of thinking about your research project been helpful in that way?
So I think I'll go ahead and start answering. For me, something like this always helps. Trying to really figure out what is actually important in your research and what do people actually need to know to understand your project. So what is the smallest piece of information that tells you the most in a way? So it definitely helped me for that. And I don't think you can ever get enough exercise or enough training to do that properly. So in that sense, that did really help me, yes.
Yes, thank you.
Me too. It helped me a lot because in my first training, I was sure that I can do this. I can present in 3 minutes, but at the end, we just filter for the best results, and it was amazing. Helped me a lot.
So Yun, do you have any take on this as well?
I think I'm not presenting very well, so I'm really thank this opportunity to present my work to every specialist into this area. So
yeah, it's always tricky part. So one thing that I was thinking, if you could go back to like just before entering the stage, what would be like your best tip to yourself before entering the stage and presenting for like 3 minutes?
For me, it's just breathe, maybe think about something else. I did practice my slide a lot before presenting, but before every presentation, I do get nervous. But then once I'm on stage, after like a minute or 2, it gets easier, which is not really helpful for a 3-minute competition where you only have 3 minutes. So yeah, really breathe, try to be in the moment, but maybe think of something else, whatever helps you. Everyone is a bit different in that. For me, it was really trying to get my mind off of the fact that I'm going to present very soon. Yes.
Yeah. We
For
can
me
go to say
was
more.
the training. I train a lot, and I think it's important to say that I'm not fluently in English, and do a presentation in English is a big challenge, and do a presentation in 3 minutes in English is a big challenge. So I think training, and the presentation should be not exactly 3 minutes, but 2 minutes and 30 seconds, something like this, because the problem with the English and the nerves, is an issue in the day. So it's like this.
Yeah.
Yeah, I'm agree with Dinah and Jenna. And my English is not very fluent, so it's very surprised that I won this prize actually.
So then I guess in conjunction to this, like I don't know, in my previous lab we had like so many people from different backgrounds and stuff. So I mean our main language of communication was like English in the lab. So how has this looked in your like labs in the past?
So my lab in Austria was, I think, 100% at times, and at other times 90% German speakers and people from Austria. So my studies in chemistry were all in German, exclusively in German. Then when I moved to Switzerland, I was part of two labs. Labs that were quite international. So there the working language was English. Sometimes we would switch to German when there were only German speakers in the room, but it was mostly English. And now in my postdoc, it's also mainly English, actually. So that does help a lot with practicing, and that does help a lot also with presentation skills.
So how is it for the other people?
It was my first presentation in English, I think. It was a challenge, but it was an amazing challenge, and I was really happy at the end.
Every time I have to present in English, I just wrote down a very detailed script and memorize it. So I'm sorry, I cannot give any advice to future competitors.
Yeah. So in conjunction to, I guess, the 3-minute thesis competition, like if you think like not about your own presentation, but like I mean you had to see like 11 other presentations as well, right? So what would you say make for the most like memorable part of like this kind of flash presentations where you don't have a lot of time to like explain your topic and stuff?
Most memorable is difficult to say. You definitely hear a lot. It's a lot of different information in a very condensed time frame. For me, I really like the people. You have a nice, friendly atmosphere there. It didn't feel like we're in a competition. It felt like whoever wins, we're gonna celebrate together, which was nice. So yeah, overall, I think that was all in all very memorable, and it was cool to just participate in something like this. I never saw a competition like this before at any conference. I don't know if there were any other competitions like this, so that was a great opportunity in and of itself.
And I think it's really interesting, the 3-minute competition, because we need to show the most important data that we have in 3 minutes. So I think the public is really interesting because it's fast and you can see the results directly. And I think that if I need to go to a competition, the 3 minutes is interesting because of this. I will see the best data in a direct way.
For me, yeah, I think it's a very interesting competition, and the atmosphere also is very, very. Overall, I think I hope that this competition can continue.
Cool. So I guess we're running into like the last minutes of the session. So I guess as for a final question, unless we have anyone in the audience who wants to ask something, I guess the main question is: will you be attending Dresden this year?
Unfortunately, I think not. I would like to. I still need to convince my PI to allow me to go. Otherwise, there are other conferences that I would attend in Germany, so I would need to prioritize. But I would love to go. I think it was fun.
And I'm going to try again. I need to convince my supervisor too. I started my my plan to convince him, but yes, it wasn't so amazing, and I would like to back and participate again now in Germany.
Because I have left the lab for a while, so I'm not very familiar with these things now. So I hope others can join this competition.
But in that case, I think I will thank all of you guys for stellar presentations and great inspiration for people attending the 3-minute thesis competition this year. And as I guess you have hinted to already, like if you want like brief presentation which provides you like a lot of insights into different areas of the field, I think it's a great competition to just observe as well as a in the audience. So hope to see you all this year in Dresden, and thank you so much.
Thank you.
And goodbye.
Thank you, everyone. See you. Bye.